We completed the plant kingdom, classification of plant kingdoms, Paragara, Rhizopus, Funaria, Pteris, Cycas. You are definite of getting one bit from each. Now today we discuss microbiology. Microbiology with bacteria and virus. Bacteria may be complete and virus will be up to symmetry of viruses. Microbiology is a science which deals with all the organisms which are visible only under microscope. For example, bacteria, virus, protozoans, algae, fungi. All these are seen only under microscope. This microbiology is called study of microorganisms previously, but a scientist by name Roger Stanier proposed that microbiology is not only the study of structure and other aspects of microorganisms, but also their artificial culture techniques in special laboratories or in laboratories in special culture media. So many aspects are there. For example, LAB is there, lactic acid bacteria, which helps in the conversion of milk into curd. There are so many bacteria from which antibiotics are obtained. For example, streptomyces is obtained from streptomyces. There is one fungi by name Penicillium notatum, which gives a wonder drug penicillin. Then bacteria forms the source of so many vitamins, amino acids and many other components. Previously, extraction of uranium, radium and rare earth metals used to cost more, but now by using microbiology or bacteria, their cost is reduced very much. Study of microorganisms in other space or other areas is called exomicrobiology. Like this, then bit cotton, Bacillus thuringiensis cotton is incorporated to cotton and thus is resistant to various insecticides. This BT, Bacillus thuringiensis, is called as, very popularly called as bioinsecticide. Then we discuss about bacteria. So many aspects, structure, number arrangement, and then nutrition in bacteria, gram staining in bacteria, pleomorphic bacteria. Then we discuss nutrition, after that reproduction in bacteria with conjugation, transformation, transduction, and also economic importance of bacteria. Bacteria useful in agriculture, bacteria useful in industry, bacteria useful in medicine, bacteria useful in biotechnology, and harmful bacteria, food poisoning bacteria, bacterial diseases in plants, bacterial diseases in animals, and bacterial diseases in human beings. After that we discuss viruses. In viruses we study about structure and many other aspects, structure of tobacco mosaic virus and other aspects. Then also we discuss replication in viruses and also transmission of viruses, viral diseases in plants, and finally how viruses are controlled. These are the chapters. Now we discuss some of the questions in bacteria and viruses. First, identify the wrong statement regarding bacteria. Invagination of plasma membrane are seen in gram positive bacteria. It is correct. What is the invagination of plasma membrane is called? Suppose this is the bacterium, it has a cell wall, then this is the plasma membrane which shows invaginations. These invaginations of plasma membrane are popularly called mesosomes. So these are found in gram positive bacteria. Mesosomes help in exchange of genetic material equally to the daughter cells, breakdown of cell after the binary fission, and distribution of cell organelles to the daughter cells. Then, so invagination of plasma membrane is seen in gram positive bacteria is correct. Teichoic acid is absent in gram positive bacteria is wrong. Pili are seen in some gram positive bacteria. There is a bacterium by name Corynebacterium renale. A bacterium by name Corynebacterium renale. This is a gram positive bacteria with pili. So Corynebacterium renale is a gram positive bacteria with pili. So pili are found in some gram positive bacteria is correct. Then flagella are absent in all coccoid bacteria. All cocci bacteria, very few bacilli bacteria, flagella are completely absent. So identify the wrong statement regarding bacteria is teichoic acid is absent in gram positive bacteria. It is a wrong statement because teichoic acid is present in gram positive bacteria, absent in gram negative bacteria. Next, pathogenic nature of bacteria on plants, animals and human beings was first observed by. Bacteria are going to cause diseases. Bacteria are responsible for diseases in plants, animals and human beings was first experimentally proved not by Whittaker and Haeckel, not by Leeuwenhoek and Ehrenberg, not by Whittaker and Ehrenberg. It is Louis Pasteur and Koch. Why? Because Louis Pasteur was the first scientist who proposed that rabies due to dog's bite is caused by some organisms which are definitely smaller than bacteria. Then Koch's proposed postulates how to culture a bacterium in the laboratories. So pathogenic nature of bacteria on plants, animals and human beings was first observed by Louis Pasteur and Koch. First option. Next. Scientists associated with the study of bacteria: Ehrenberg, Stanley, Louis Pasteur, Robert Koch, Ivanoski and Haeckel. Ehrenberg coined the term bacteria. Louis Pasteur, popularly known as pasteurization experiments, heating milk and other liquids at about 62.3 degrees centigrade in order to eliminate all harmful microorganisms, is called as pasteurization. Conversion of anaerobic bacteria into aerobic bacteria by supply of oxygen externally is called as Pasteur's effect. So, whenever Pasteur name comes, he was related with bacteria and he was very popular, regarded as father of bacteriology. So, Ehrenberg, Louis Pasteur, Robert Koch. Robert Koch proposed postulates regarding this, and Haeckel. Haeckel's name is also associated with bacteria. So, scientists associated with the study of bacteria are Ehrenberg, Louis Pasteur, Robert Koch and Haeckel. Study the following table and identify the correct one. Cell wall muramic acid prevents desiccation is wrong. Cell wall muramic acid present in the cell wall is correct, but preventing desiccation is done by capsule, not by cell wall. Slime layer polysaccharide antibiotic resistant. Slime layer is made up of polysaccharides, okay, but it is not giving any antibiotic resistance. It makes the bacterial cell to get attached to hard objects present in the water, plants and other things. Capsule polysaccharide prevents desiccation. Capsule is present above the cell wall. Actually, it is called glycocalyx. Glycocalyx in the initial stages is a liquid like form called slime layer. Slowly slime layer becomes a little bit thick layer called capsule. Then capsule becomes very much thick layer called S layer. All these three combinedly form glycocalyx. So, here cell wall muramic acid prevents desiccation is wrong. Slime layer polysaccharide antibiotic resistance is wrong, but capsule polysaccharide prevents desiccation. Capsule contains maximum water. It prevents the desiccation of drying up of bacterial cell. So, only 3 is correct. This is an assertion reasoning question. Some microbes help in creating disease-free world. Some microbes help in creating disease-free worlds. Some microbes produce antibiotics and vaccines which are used in curing the disease or protecting us from getting diseases. Some microbes, Streptomyces griseus gives streptomycin and cycloheximide. Penicillium notatum gives penicillin. So, like this, so many antibiotics are chemicals obtained from bacteria are useful in creating a disease-free world. Like that, some microbes help in creating disease-free world is correct. How they are maintaining a disease-free world is reason. Some microbes produce antibiotics and vaccines which are used in curing the diseases or protecting us from diseases. So, this is A and R are correct. R is the correct explanation of A. Now, this is a matching column. Study the following list and identify the correct matching. Cristispera, Beggiotova, Lactobacillus, Escherichia coli, gram negative, spiral, filamentous, spherical, gram positive. Cristispera is spiral. Cristispera and Spirocheta are spiral chain-like bacteria. Beggiotova is filamentous. Beggiotova is long filamentous special bacterium. It appears like threads or filaments. Lactobacillus. Lactobacillus is a gram-positive bacterium which helps in the conversion of milk into curd. Escherichia coli. Escherichia coli is a gram-negative bacterium. So, Cristispera is spiral. Beggiotova is filamentous. Lactobacillus is gram-positive. E. coli is gram-negative. So, study the following list and identify the correct matching is F2, B3, C5, D1. A. Tricus bacteria among the following. What is A. Tricus bacteria? Based on the flagella number and other things, bacteria are divided into different categories. One is A. Tricus bacterium. The other is Monotricus bacterium. Third is Lopotricus bacterium. Fourth is Amphitricus bacterium. Last is Peritricus bacterium. These 5 types based on the number of cells. A. Tricus bacterium without any flagella. flagella are completely absent. Monotrichous bacterium, only one flagella. Lopotrichous bacterium, bunch of flagella only on one side. Amphitrichous bacterium, bunch of flagella on both sides. And peritrichous bacteria, flagella all over the body of bacterial cell. So, this is the distribution according to number and arrangement or nature and arrangement of flagella. Atrichous without bacteria, without flagella. Monotrichous single flagellum. Lopotrichous bunch of flagella on one side. Amphitrichous bunch of flagella on both sides. And peritrichous bacteria with flagella throughout the body. So, atrichous bacteria among the following is Thiomargarita namabiensis is a spherical bacteria which does not possess any flagella. Epilopysium physiolsoni is the largest rod-shaped bacterium which is obtained from the gut of a special fish called Acanthurus nigrofuscus. A special fish called Acanthurus nigrofuscus gut gives rise to the largest rod-shaped bacterium Epilopysium physiolsoni. This is also a atrichous bacterium. Streptococcus is also a atrichous bacterium because all cocci circular bacteria do not possess flagella. So, atrichous bacterium among the following. Thiomargarita namibiensis is said to be the largest spherical bacterium which is obtained from ocean sediments of Namibia. From the ocean sediments of Namibia, we have discovered this Thiomargarita namibiensis. Epilopysium physiolsoni is the largest rod-shaped bacterium which was discovered from the gut of a fish called Acanthurus nigrofuscus. And Streptococcus is a spherical bacterium. So, 4 A, B, C, all the 4 is correct. Arrange the following based on their length. Arrange the following based on their length. Pili is a little bit smaller, slightly larger than pili, sex pili, and definitely larger is flagella. So, pili is slightly shorter, sex pili when compared with pili it is larger, and flagella are still larger. So, answer is B, C, A. Pili helps in attaching the bacterial cell to some hard objects. Sex pili is useful in the formation of conjugation canal during conjugation. So, answer is pili, sex pili, flagella based on their length. Next. Identify the statements among the following and choose the correct one mostly. Mesosomes help in the formation of daughter cells during binary fission. Definitely. Mesosomes are the components which cause a ridge or cleft or cell plate like formation in between two daughter nuclei and resulting in the formation of each cell into two cells. In gram-positive bacteria, infolding of cell membrane are called mesosomes. It is also correct. Mesosomes help in chromosomal replication and its equal distribution to daughter bacterium cells. Mesosomes definitely help in chromosomal replication and its distribution to special daughter bacterial cells. Mesosomes increase the absorption of nutrients. Mesosomes, because of their foldings, they increase the absorption of nutrients. So, mesosomes help in the information of daughter cells during the binary fission. In gram-positive bacterium, infoldings of cell membrane are called mesosomes. Mesosomes help in chromosomal replication and their distribution to the daughter bacterial cells. Mesosomes increase the absorption of nutrients. All are correct. So, answer is 1, 2, 3, 4. Now, these are a matching column. Animalcules, penicillin, streptomyces, thiomargarita. Animalcules always by Leeuwenhoek. Penicillin by Alexander Fleming. Streptomycin is by Waxman. Thiomargarita is by Hedi Schulze. Hedi Schulze was the scientist who discovered thiomargarita. So, animalcules, the term animalcules was given by Leeuwenhoek, which later converted into bacteria by Ehrenberg. Penicillin is extracted by Alexander Fleming, a wonder drug. Streptomycin is always by Waxman and thiomargarita is by Hedi Schulze. So, the matching is third one, A2, B3, C4, D1. This is about matching question. Choose the correct statements in the following. Bacteria are included in the single class called Schizomystis. Why they are kept in Schizomystis? Because the main method of reproduction is binary fission. That's why bacteria are included in a single class called Schizomystis. Bacteria chromosomes has no histone proteins. So, this is also correct. Bacterial chromosome is circular and does not show any histone proteins. The characteristic mode of reproduction in bacteria is binary fission. So, always the maximum method of reproduction in bacteria is binary fission. The cell wall of gram-positive bacteria contain teichoic acid. The cell wall of gram-positive bacteria contain teichoic acid. So, bacteria are included in a single class called Schizomystis is correct. Bacterial chromosome has no histone proteins is correct. The characteristic mode of reproduction in bacteria is binary fission is correct. The cell wall of gram-positive bacteria contain teichoic acid is correct. So, answer is 1, 2, 3, 4, fourth option. This is an assertion reasoning question. Azotobacter is a pleomorphic bacterium. Azotobacter is a pleomorphic bacterium is wrong. Pleomorphism means a bacteria changing its shape according to nutritional requirements and environmental effects. Pleomorphic bacteria is Acetobacter. This Acetobacter changes its shape according to nutritional requirements and environmental conditions. So, pleomorphic bacteria or a bacterium that exhibits pleomorphism is Acetobacter, not Azotobacter. So, assertion is wrong. Pleomorphic bacteria change their shape depending upon the type of environment and nutrients available. So, this is correct. Pleomorphic bacteria always change their shapes depending upon the type of environment and nutrients available. So, answer is A is false, R is true. Find out the correct feature related to lactic acid bacteria lab, LAB. Grow in milk and convert into curd. It is correct. Improve nutritional quality by increasing vitamin B12. This is also correct. It increases cyanocobalamin or vitamin B12 to increase the nutritional quality. Checking the disease causing microbes. So, it also controls the disease causing microbes. So, find out the correct features related to lactic acid bacteria LAB is grow in milk and convert it to curd is correct. Improves nutritional quality and by increasing the vitamin B12 or cyanocobalamin is correct. Checking disease causing microbes is correct. So, answer is 1, 2 and 3. So, third option. This is an assertion and reasoning question. Assertion. Bit cotton is resistant to attack by insect pests. Bit cotton is nothing but a cotton infested with Bacillus thuringiensis. A cotton infested with Bacillus thuringiensis is BT cotton or simply called as bit cotton. By genetic engineering methods, the scientists have introduced Bacillus thuringiensis toxins genes into plants. BT cotton or Bacillus thuringiensis is resistant to attack by insect pests is correct. How it entered? By genetic engineering methods, scientists have introduced Bacillus thuringiensis toxin genes into plants is correct. So, both A and R are correct. R is the correct explanation of A. First option. In the following member, Alexander Fleming discovered antibiotic. Alexander Fleming name associated with penicillin. Penicillin is obtained from Penicillium notatum. Penicillin is obtained from Penicillium notatum. Penicillium notatum is a fungi belonging to a special class called Ascomystes. So, Penicillium notatum is the fungi from where Alexander Fleming discovered penicillin, and Penicillium notatum is a fungi. So, the question is a simple question. In the following member, Alexander Fleming discovered antibiotic. Not algae, not bacteria, not yeast, it is fungi, Penicillium notatum. This is an assertion reasoning question. E. coli, Escherichia coli is a gram-negative bacteria. This is correct. Teichoic acid is absent in the cell wall of gram-negative bacteria. So, E. coli is a gram-negative bacteria, not because only the character that teichoic acid is absent in cell wall of gram-negative bacteria. E. coli as a gram-negative bacteria is correct. Teichoic acid is absent in the cell wall of gram-negative bacteria is correct. But the special reason is not because of the teichoic acid absent in the cell wall, the gram-negative nature of E. coli is decided. There are so many other features also. So, both are correct, not related. Hence, A and R are true. R is not the correct explanation of A. Second option for this question. Now, study the following table: structure, composition, function. Capsule, polysaccharides, immunogenic. Here we have to identify incorrect one. Capsule, polysaccharides, immunogenic is correct. Plasma membrane lipoproteins protection from desiccation is wrong. Cell wall peptidoglycan pathogenic nature is correct. So, in this 1, 2, 3, which one is wrong? Capsule made up of polysaccharides, it is immunogenic is correct. Plasma membrane made up of lipoproteins is perfectly correct, but it is not giving protection from desiccation. It is not at all involved with desiccation factor or something like that. Cell wall peptidoglycan. Cell wall is made up of peptidoglycans or muramic acid or murines, which gives pathogenic nature. So, 1 and 3 are correct. Second is wrong. So, answer for this incorrect is 2 only. Now, this is a assertion reasoning question. Rhizobium grows as a symbiont in the roots of legume plants. We all know biofertilizer rhizobium inoculants means rhizobium. Rhizobium lives in the root nodules of legumes. Help in nitrogen fixation is a special feature of all legumes. What you call all rhizobium bacteria which enter leguminous roots and form root nodules and help in nitrogen fixation. Reason: all bacteria acts as symbionts. All bacteria acts as symbionts is wrong. Some are pathogenic, some are saprophytic, some are parasitic. So many types of bacteria are there. You cannot simply say that all bacteria acts as symbionts when rhizobium grows as a symbiont in the root nodules of legume. So, rhizobium grows in the root nodules of legumes is correct. All bacteria are symbiotic is wrong. So, A is true, R is false. Involutions of cell membrane of a bacteria are known as. Involutions means the folding inside. Foldings inside in the bacterial cells are not called as ribosomes. They are not called as lomosomes. They are not called as plasmids. Ribosomes are 70S ribosomes present as polysomes. Lomosomes are not found in bacterial cells. Plasmids, extra chromosomal genetic material, circular chromosomal genetic material with some special properties are plasmids, with R plasmid, P plasmid, F plasmid and coli plasmid. The answer: involutions of cell membrane of bacterium are popularly known as mesosomes. Third option. Gas vacuoles are commonly seen in. Gas vacuoles are special vacuoles in some bacteria. Gas vacuoles are special vacuoles in some bacteria which are impermeable to water but permeable to gases. Permeable to gases. These make the bacteria to float on water. These make the bacteria to float on water. So, gas vacuoles are very important. These gas vacuoles are impermeable to water. They do not allow water to go inside, but they are permeable to gases. They allow the gas to pass through. Chromatium, chlorobium, nitrosomonas. Chromatium, chlorobium shows gas vacuoles. Nitrosomonas does not show gas vacuoles. So, answer is 1 and 2 are correct. Arrange the following in a sequence from periphery towards the center of a bacterial endospore. Every cell produces one endospore during unfavorable conditions. In this endospore, the outermost layer is called spore coat. Inner to spore coat, there is a layer called cortex. Next to cortex, plasma membrane is present, and finally spore wall is present. So, outermost layer is spore coat. Next to spore coat, there is a cortex. Next to cortex, there is, so first is spore coat, then is spore wall, then is cortex, and finally plasma membrane. So 3, 2, 1, 4. What is 3? Spore coat. What is 2? Cortex. What is 1? Spore wall, and 4 plasma membrane. 3, 2, 1, 4 in center, from towards the periphery to center. Periphery to center. Arrange the following structures in bacterial center from periphery to center. Bacterial endospore, periphery to center, fourth one. 4, 2, 1, 3. What are the exosporium, spore coat, cortex and spore wall? So, same question, a little bit exosporium is additionally given. Now, bacterium host disease. Bacterium host disease. Erwinia, host is animal actinomycetes. Treponema, man syphilis. Bacillus, animal anthrax. Corynebacterium, plant diphtheria. So 2 and 4 are 2 and 3 are correct combinations. Treponema in man syphilis, Bacillus animals and anthrax, and Corynebacterium plant diphtheria is wrong. Erwinia animal actinomycetes is wrong. This is regarding antibiotics obtained from various bacteria. Cycloheximide, oxytetracycline, amphotericon, neomycin. Cycloheximide is from Streptomyces griseus 5. Oxytetracycline is from Streptomyces rhinosus. Streptomyces rhinosus. Amphotericon is from Streptomyces nodosus, and neomycin is from Streptomyces fradiae. So, cycloheximide is from Streptomyces griseus. Cycloheximide and streptomycin both are obtained from Streptomyces griseus. Oxytetracycline is obtained from Streptomyces rhinosus. Amphotericon is obtained from Streptomyces nodosus. Neomycin is obtained from Streptomyces fradiae. So, A4, B1, C2, D3. This is the correct combination. So, what are the Bacillus laecalyformis is giving bacitracin. So, no bacitracin is given in that side. So, cycloheximide, Streptomyces griseus, oxytetracycline, Streptomyces rhinosus, amphotericon, Streptomyces nodosus, neomycin, Streptomyces fradiae. This is various antibiotics obtained from bacteria. Match the following, identify the correct match. Clostridium falcineum. Clostridium falcineum is retting of fiber. Clostridium butyricum and Clostridium falcineum helps in retting of fiber. Streptomyces nodosus is amphotericon. Amphotericon. Brevibacterium or Brevibacterium or Brevibacterium or whatever it may be, it is giving single celled proteins. Micrococcus. Micrococcus also used in curing tea. Bacillus megatherium is used in curing tobacco leaves. Micrococcus helps in curing tea. So, Clostridium falcineum, retting of fibers. Streptomyces nodosus, amphotericon. Brevibacterium, single celled protein. Streptomyces venezuela gives chloramphenicol, that is not given. Micrococcus is used in curing tea leaves. So, second option. Lederberg and Totem discovered conjugation in bacteria, particularly in Escherichia coli. There are two bacteria in this, gram positive, gram negative. Gram positive is called F plus, gram negative is called F minus. In gram positive there is a plasmid and this plasmid replicates. In gram negative there is, in F plus plasmid is there, in F minus plasmid is not there. F plus plasmid replicates and this plasmid passes through the sex pili conjugation canal, enters the female cell and converts the female cell into male cell. So, this is conjugation. It is the only physical contact. In transformation and transduction there is no physical contact. So, only physical contact by which reproduction takes place in bacteria is conjugation. Lederberg and Totem discovered conjugation in Escherichia coli. The bacterium used in retting of Sunhem. Bacillus is used in ammonification. Brevibacterium or Brevibacterium is used as a single cell protein. Micrococcus is used in different purpose, but now the bacterium is used in retting Sunhem is Clostridium falcineum and Clostridium butyricum. Two bacteria are widely used in retting of Sunhem. What is retting? Extraction of fiber from the plant is called as retting. Brevibacterium is a bacterium. Brevibacterium is a bacterium used in lysine production. Lysine production is given by Corynebacterium glutamicum. So, Corynebacterium glutamicum gives lysine. Ethanol is given by Zymomonas mobilis, or Thermoanerobacterium ethanolicus. Bioinsecticide. We all know, just now we discovered about bioinsecticides, Bacillus thuringiensis. So, Corynebacterium glutamicum gives lysine. Ethanol is obtained from Zymomonas mobilis, Thermoanerobacter ethanolicus. Bioinsecticide bacterium is Bacillus thuringiensis. So, question is Brevibactum. Brevibactum is a bacterium which is used as a single-celled protein. Pastorella causes the following diseases in human beings. Pastorella pestis always causes plague. Leprosy is caused by Mycobacterium leprae. Typhoid is caused by Diplococcus or Salmonella typhi or Salmonella typhimurium. Cholera is caused by Vibrio cholera. So, plague is caused by Pastorella pestis. Leprosy is caused by Mycobacterium leprae. Typhoid is caused by Salmonella typhi. Cholera is caused by Vibrio cholera. So, Pastorella causes the following diseases in human beings is plague. Pseudomonas causes wilt disease in Solanaceae plants. Wilt in Solanaceae plants is always caused by Pseudomonas solanacearum. So, wilt in Solanaceae plants is caused by Pseudomonas solanacearum. So, Pseudomonas causes wilt disease in the members of not Fabaceae, members of not Malvaceae, members of not Asteraceae, members of Solanaceae. So, Pseudomonas causes wilt disease in the members of Solanaceae. Blight of rice is caused by citrus canker, angular leaf spot in cotton, and blight in rice. All are caused by single genus Xanthomonas. Citrus canker is caused by Xanthomonas citri variety, Xanthomonas citri. Angular leaf spot is caused by Xanthomonas malvacearum. Blight in rice is caused by Xanthomonas oryzae. So, citrus canker is by Xanthomonas citri. Excellent. Blight of rice is caused by Xanthomonas oryzae. Erwinia amylovora cause fire blight in apples. Pseudomonas solanacearum, Pseudomonas and many other things are there. Agrobacterium tumefacius causes crown gall in apples. So, these are the answer is 1. HFR strains formed during bacterial conjugation. HFR means high frequency plasmids. Here the plasmid is incorporated into the DNA of bacterial cell. During replication, two cells come close. During conjugation, two cells come close and sexpili form conjugation canal. Immediately HFR strain undergo replication and enters the other cell either completely or partially. That is why the zygote formed is called partial zygote. So, binary fission is wrong, transformation is wrong, transduction is wrong, answer is conjugation. The substance that helps in the integration of single stranded DNA fragment into bacterial chromosome during bacterial transformation, not exonuclease. Exonuclease only cuts the DNA into small bits, not endonuclease, not DNA binding proteins, but it is competence of specific protein which makes the DNA to enter bacterial cell and fuses with the bacterial DNA or host bacterial DNA in an inherited form. So, the substance that helps in the integration of single stranded DNA fragment into bacterial chromosome during bacterial transformation is competence specific protein. Viruses are involved in one of the following methods of bacterial reproduction. Viruses are involved in a special method called transduction. What is transduction? Transduction was reported in Salmonella typhimurium by Zinder and Lederberg. What is transduction? The transfer of bacterial DNA from one bacteria to other bacteria with the help of bacteriophage is called transduction. The movement or transfer of genetic material from one bacteria to other bacteria with the help of a bacteriophage is called as transduction. So, lytic cycle is wrong, lysogenic cycle is wrong, transformation is incorporation of foreign DNA from the surroundings into the bacterial cells. So, answer is transduction. Which of the following is correct sequence events during binary fission? New cell wall is laid, appearance of constriction, invagination of plasma membrane, DNA replicates. First, replication of DNA takes place, then invagination of plasma membrane occurs, then appearance of constriction occurs, then new cell wall is laid. It is correct sequence. DNA replication, invagination of plasma membrane, appearance of constriction, and new cell wall is laid. So, third one, 4, 3, 2, 1. So, this is a matching question again. Blight of rice is by Xanthomonas oryzae. So, 1 is D. Citrus canker is by Xanthomonas axanopodis PV, pathogenic variety citri. Xanthomonas axanopodis, pathogenic variety citri. Fire blight of apples, Erwinia amylovora, wilt in tomato, Pseudomonas solanacearum. Fire blight of apples, that apple is included here, Erwinia mistakenly. So, fire blight of apples. So, blight of rice is Xanthomonas oryzae. Citrus canker is Xanthomonas axanopodis PV citri. Fire blight of apples is Pseudomonas solanacearum, and wilt of tomato is Pseudomonas solanacearum. Fire blight of apples is Erwinia amylovora. So, AD, 1D, 2A, 3B, 4C. Bacteria used in retting of sunham. Again, extraction of fiber, Clostridium falcineum and Clostridium butyricum. Clostridium falcineum, Clostridium butyricum, Clostridium tetani, Clostridium acetobutylicum. So, Clostridium falcineum, Clostridium butyricum, 1 and 2 will be the answer. Clostridium tetani causes tetanus. Clostridium acetobutylicum forms the source of acetone and butanol. So, answer is 1. This is a assertion reasoning question. Some bacteria are treated as scavengers of nature because they clean the atmosphere. Why they are called as scavengers of nature? Because dead bodies of plants and animals are decomposed by some bacteria. Because the dead bodies of plants and animals are decomposed by bacteria, they are called scavengers of nature. So, A and R are true. R is the correct explanation of A. So, some bacteria are treated as scavengers of nature is correct. Why they are called scavengers of nature is reason. Dead bodies of plants and animals are decomposed by some bacteria, saprophytic bacteria. A and R are true. R is the correct explanation of A. In biotechnology, Agrobacterium tumefaciens plays a very important role. So, in biotechnology, Agrobacterium tumefaciens called TI plasmid tumefaciens. Agrobacterium in tumefaciens is called TI plasmid, that means tumor inducing plasmid. So, in biotechnology, Agrobacterium tumefaciens plays a very important role. It is used as a vector in genetic engineering. This TI plasmid is very popular vector in producing clones. Biotechnology, clones. So, both A and R are correct. R is the correct explanation of A. So, in Agrobacterium tumefaciens, Agrobacterium tumefaciens cause fire blight in apples. Fire blight in apples. And special feature is it has TI plasmid. What is TI plasmid? Tumor inducing plasmid. Tumor inducing plasmid. This tumor inducing plasmid is having the capacity of getting foreign DNA and showing maximum replication. Whenever plasmid undergoes replication along with foreign DNA, same copies of genes are produced, which is called as clones. So, Agrobacterium in biotechnology, Agrobacterium tumefaciens plays a very important role is correct. Why it plays an important role means it is used as a vector in genetic engineering is correct. So, both A and R are correct. R is the correct explanation of A. Next, another assertion. Symbiotic bacteria are pathogenic in nature. Symbiotic bacteria are pathogenic in nature. Escherichia coli is a common symbiont found in human intestine. Symbiotic bacteria pathogenic nature is wrong. Symbiotic bacteria pathogenic in nature is wrong, because all symbiotic bacteria are very much useful. For example, Rhizobium, it is useful in nitrogen fixation by their living in root nodules of legumes. Rhizobium contains that specific enzyme called nitrogenase. Nitrogenase breaks the triple bond, strong triple bond between molecular nitrogen present in nature. So, symbiotic bacteria are pathogenic in nature is wrong. Escherichia coli is a common symbiont found in human intestine is correct. So, all symbiotic bacteria are not pathogenic, they are useful. What is symbiosis? A close association between two completely different individuals where both are getting benefited is symbiosis. So, symbiotic bacteria are pathogenic nature is wrong. Escherichia coli is a common symbiont found in human intestine is correct. So, A is false, R is true. So, these are some of the questions in bacteria, important questions. Now, we go to viruses. Viruses up to symmetry. Virus is acellular organism. Acellular means it don't have any life. Outside the host it is non-living and crystalline. Inside the host they cause lot of diseases. There are some non-living characters and some living characters. What are non-living characters? They don't show a complete identity. They don't have any life outside the host. 2. They don't have protoplasm and nucleus. 3. They are outside the host, completely non-living and crystalline. These are some of the non-living characters. Coming to the living characters, once they enter the host, they cause diseases. Then they show genetic continuity, and above all, they show response to mutations. Because of this, they are said to be living. So, viruses are connecting link between living and non-living components is the general statement. Viruses are nothing but poison. Virus means a poison. No virus is useful. Even common cold is unbelievable transmittable diseases from 1% to 10%. Viruses are discovered first by Louis Pasteur, who discovered rabies is caused by some organism, not bacteria. Later, Edward Jenner discovered smallpox vaccination. Then, Adolf Meyer discovered tobacco mosaic disease is caused by some organism smaller than bacteria. Those small particles are called as filterable agents by Ivanoski. Then Contagium vivum fluidum by Bisering, he called the term virus. First tobacco mosaic virus is obtained in a pure crystalline form by W.M. Stanley. Chemical nature of viruses was proved by Bowden and Pyrie. Franklin Conrad proved that RNA is the genetic material in plants. Then Shearer and Schramm proposed that virus contain genetic material which is not only genetic material but also a pathogenic component. So, like this, lot of history, so many important aspects. Now, we discuss some questions. First, the non-living character of virus is absence of RNA is wrong, absence of DNA, virus shows either DNA, RNA, presence of inorganic crystals, definitely it is present as per inorganic crystal outside the host, no response to external stimuli, irritability is completely absent in viruses. So, irritability is the primary character of almost all living components. So, because it does not show any response to external stimuli, this is a non-living character of viruses. So, fourth option. Edward Jenner discovered. Edward Jenner is a very popular scientist who proposed that smallpox disease is caused by some organism which is definitely smaller than bacteria. By discovering the smallpox vaccination, he also discovered a vaccination for smallpox. So, smallpox virus, rabies virus, vaccination for rabies is by Louis Pasteur, vaccination for smallpox is by Edward Jenner. The existence of disease causing organisms smaller than bacteria was indicated by Ivanoski. Ivanoski is a scientist who took the sap of mosaic infected leaves of sap from mosaic infected leaves of tobacco. He allowed this sap to pass through a filter paper. Filter paper shows minute pores through which even bacteria cannot pass, but the particles present in the sap are easily moving through these pores. Hence, he called them as filterable agents. So, he called those particles present in the sap of mosaic infected leaves of tobacco as filterable agents. So, the existence of disease causing agents smaller than bacteria was indicated by Ivanoski as filterable agents. The distinct nature of virus was established by Bisering, very popular man. He took the sap of mosaic infected leaves of tobacco. He took the sap of mosaic infected leaves of tobacco and rubbed it to healthy leaves. Surprisingly, healthy leaves also got disease. That's why he called that sap as Contagium vivum fluidum, which is nothing but contagious living fluid. With this term Contagium vivum fluidum, he proved the particles present in the sap are causing the disease. Because they are poisonic in nature causing the disease, he coined the term virus. So, Peary, chemical nature of viruses along with Boden, Gierer along with Schramm proved that the genetic material of virus is not only a genetic material but also cause diseases. Protein component of virus was first discovered by. Protein component of virus means viruses are basically made up of nucleoproteins. They show a nucleic acid either DNA or RNA, then they are surrounded by a protein coat called capsid protein coat. So, Boden and Peary proved the protein nature of viruses. So, Schramm, Franklin Conrad proved the genetic nature of RNA in plant viruses. Gierer and Schramm, just now I told, genetic material of virus is not only genetic material but also infectious agent. So, fifth question answer is 1.